Yeah, so my name is Cammy and I'm a postdoc in Kelly Watson's lab, and I'm currently working on farmed Pekin ducks. So today I'll be talking through an analysis I did on some Pekin, on some pedigree mating data, which was rooted in some ideas in behavioural ecology. And it was a project I did in collaboration with Cherry Valley Farms and NTNU in Norway.
Sorry, just wanna. It seems like they can't hear us. Minu, is that true? You can't hear us right now? No. Let's try here with our settings. Devices. It is on our webcam. So if anyone online can hear us or not, no, yes or no?
Yes, we can hear you.
Oh, perfect, thank you. Sorry, continue.
So just
you need to unmute, Cammy, for us to hear.
I'll send her a message.
We still cannot hear you.
Okay, I think Chrissy's sending you a message, Minu. I'll just crack on.
Yeah, just keep going. I'm sending Minu a message right now.
Yeah, so just a quick introduction to myself. So I have a PhD. So I obtained my PhD in behavioural ecology from the University of Edinburgh, and that was from the Institute of Ecology and Evolution. And at the end of my PhD, I really wanted to explore ways that I could integrate concepts from behavioural ecology into a more applied research questions. So this led me to join Kelly's lab group in 2021, which was a collaboration between Roslyn and Cherry Valley Farms. And specifically the role was to look at how we could improve reproductive performance in commercial breeding flocks. And I saw lots of opportunities to use a behavioural perspective here. So as part of my postdoc, I work with a leading duck breeding company, so Cherry Valley Farms, and they supply breeding stock to the global duck meat industry. And their breeding program focuses on natural matings as opposed to artificial insemination. So it provides the opportunity to study really complex social interactions. So the breeding setup involves groups of housed males and females, which are grouped together, complete with nest boxes so they can lay their eggs. And this environment allows for plenty of complex social interactions such as courtship, competition, and mate choice. And this will allow us to understand their reproductive behaviour in a more sort of controlled but natural setting. So the data I work with includes behavioural data, reproductive data, and also genomic information, which you have on the birds. But today I'll just be focusing on the reproductive and genomic data which we have. So chick production is really a key part of farming Pekin ducks, and this is because there's a demand for day-old chicks in the commercial duck breeding industry. So to maintain high rates of fertilization, maintaining high rates of fertilization and therefore the number of chicks is important to improve commercial productivity. But in commercial duck farming, there's a real challenge of low fertility levels. So in particular, of the eggs which end up being incubated, about 22% of these eggs will end up being infertile. And as you can imagine, this is a huge source of waste for the duck breeding industry, which they would like to solve. So how could we try and increase chick production? Well, we could try and take a behavioural ecology approach. So behavioural ecology investigates the evolutionary basis for animal behaviour in response to ecological pressures. So it examines why animals behave the way they do, with a focus on environmental factors such as social interactions, and how these contribute to an organism's fitness, so their ability to survive and reproduce. So we can use a behavioural ecology approach to identify adaptive behaviours that impact breeding efficiency and success. And in the context of this specific problem, we can frame chick production in the context of reproductive strategies and male/female fitness. And we can combine concepts from behavioural ecology with other relevant areas of biology, such as quantitative genetics and animal breeding, to answer this question. So from behavioural ecology perspective, we can ask what behaviours increase reproductive success. From quantitative genetics, we can ask how heritable are these behaviours. And then from animal breeding, we can ask, or we can use genetic tools to help us improve the breeding population. And one reason why commercial chick production really benefits from taking this behavioural ecology approach is that, as I was saying, it relies on natural matings. So these open breeding pens consist of a 1:4 ratio of males to females, with typically about 20 to 25 males and 100 to 125 females. And we can investigate all of these complex social interactions in this environment. So this brings me onto the data set that I was working on. So Cherry Valley provided a historical data set from one of their genomic breeding lines. And this enabled me to look at the breeding interactions within a pen, which resulted in viable offspring. So for example, I could look at the number of mating partners which each male and female had, as well as the number of offspring of each individual. And as it was the pedigree from these pens also allowed us to look at the reproductive parameters for every single individual. And of course, because it was pedigree data, it didn't just include data from one pen, but we had multiple pens and data that spanned over 2019 to 2022. And this led to quite a good data set, including data from 8 breeding pens with a number of breeding adults and chicks in our data set. So to help me analyse this data, I sought a collaboration with a group of researchers from NTNU in Trondheim, Norway. So they were Dr. Yimen Aria Ahoy and Professor Jonathan Wright. And both Yimen and Jonathan are behavioural ecologists who work with wild house sparrow populations. So they're really experts in asking how different reproductive strategies can confer fitness benefits and how this may affect individuals' ability to respond to selective pressures. And I funded this project in collaboration with through an external funding network called the SQUID collaboration. So this stands for the Statistical Quantification of Individual Differences, and it's essentially a group of behavioural ecologists who take a quantitative genetic approach and want to provide PhD students and postdocs with the opportunity to collaborate with a wider group of researchers. So I travelled to Trondheim, Norway between February and March in 2023. And it was a great opportunity to live and experience a different culture, but it also provided us with a great opportunity to brainstorm different hypotheses as to what could be occurring in our data set. And these in-person discussions were really valuable. And as you can see, most of these discussions were mainly done with drawing blackboard on chalk on blackboard. So the hypothesis that we quickly came up with that could be tested with our data set was the sperm limitation hypothesis. So the sperm limitation hypothesis predicts that chick production is dependent on females receiving sufficient sperm in order to fertilise all of their eggs. And when females receive sufficient sperm, then chick production should be high. But when females receive insufficient sperm, either through lack of mating opportunities or because males simply can't provide enough sperm, then chick production will decrease because she doesn't have enough sperm to fertilize all her eggs. And there are quite a few mating strategies which we can hypothesize will be associated with the sperm limitation hypothesis. So first of all, if females are sperm limited, then one way that they can increase their availability of sperm is to increase the number of mating partners in order to just maintain more sperm that way. So we might expect female polyandry to increase in response to sperm limitation, and therefore we should expect an increase in chick production in response to female polyandry. But an increase in female polyandry may also increase a male's exposure to sperm competition. So it may not be in a male's interest to mate with a highly polyandrous female. So males can attempt to monopolize females, preventing other males from mating with her. But this male monopolization can be great for the male, but it will exacerbate sperm limitation issues in the female. And so we might expect that an increase in male monopolization will result in a decrease in chick production. And then thirdly, we might also expect the number of mating partners that a male has to impact female chick production. So if sperm production is quite costly, then males that mate with a really high number of females may become sperm depleted over time. And so females which mate with males which they themselves have a lot of mates will exacerbate this issue of sperm limitation. So we might expect high levels of male polygamy to be associated with a decline in dam chick production. So there's quite, do you have a question there?
I know some birds do it, and I feel ducks are amongst them. Does the female not choose to reject certain sperm from a male if she's not entirely happy with them?
Yeah, so chickens do that. I'm coming to that point a bit later on. I don't have evidence for it a bit, but I kind of speculate later on just to that very point, basically. Spoilers. And we can use this pedigree data to obtain measures of reproductive success in response to these mating strategies in order to test these different components of the sperm limitation hypothesis. So first of all, we need to have a measure of female reproductive success, which is just the total number of chicks that female produces. So for example, in this diagram here, we can see we have a female who has a total of 10 chicks across a variety of mating partners. However, females 2 and 3 have a lower number of chicks, in a total 8 each. Therefore, female 1 has higher reproductive success than females 2 and 3. Next, to test our impact of female polyandry on the sperm limitation hypothesis, we looked at the total number of mates each female had. So for example, for this first female, she had a total of 4 successful matings with 4 males. The second female also had 4 successful matings with 4 males, but the third female only had 2 successful matings with 2 males. So we can say that females 1 and 2 show a higher level of polyandry compared to female 3. And now, in response to high levels of polyandry, we then might expect males to start to try and monopolize females to limit their exposure to sperm competition. So as a measure of this male monopolization, we needed a metric for some kind of reproductive skew. And this measure of skew really needed to reflect the proportion of a female's brood which ends up being sired by just a single male. So for this, we use the coefficient of variation in chick paternity, which is essentially just the standard deviation divided by the mean. So for each female, we calculated this measure of reproductive skew as the standard deviation, the number of chicks sired per male she mated with, divided by the mean number of chicks sired across all the males she mated with. And this provided this relative measure of a sample's distribution, similar to sort of the standard deviation, really. But because it was a relative measure, it allowed us to compare across different females in a standardized way. So essentially, higher reproductive skew means that a greater proportion of a dam's brood is sired by a single male, whereas lower reproductive skew means that paternity is more even amongst males. So we go back to our example. Female 1 would have a high measure of reproductive skew because most of her chicks are sired by a single male. But in contrast, females 2 and 3 would have low or zero skew because chick paternity is shared equally amongst different males. So female 1 we would say is more monopolized than females 2 and 3. And we chose this coefficient of variation in chick paternity as our measure of skew because as I was saying, it provides this relative measure. So unlike a measure such as variance or simply standard deviation in chick paternity, we find, let me just word this in the correct way, essentially something like just measuring the variance in chick number across paternity. What that causes is we find that skew increases proportionally to the number of mates that a female mates with. And this confounds the relationship between skew and mating behaviour. So if we take something like the coefficient of variation, it means that whilst females 1 and 2 have the same number of mating partners, they have very different skew. And so it allows us to separate these two components. And then finally, if males have lots of mating partners, then we might expect them to become sperm depleted over time. And that could mean that a high rate of male polygamy is associated with a decline in chick production. So to have a measure of male polygamy, we just looked at the number of the total number of sire mates for dam's primary mating partner. So if we just go back to our example, female 1, her primary male mated with 3 other females, but females 2 and 3, their primary male mated with 2 and 1 females respectively. So female 1 mated with a male with a higher rate of polygamy than female 2, and then so on. Okay, so just to address how mating behaviour influenced dam production, we used an animal model. And so our response variable was the number of chicks produced per dam, which we use as our measure of reproductive success. And then our model included both a fixed effect structure and a random effect structure. So I'll just start by explaining what the random effect structure is, and then I'll go through what we included as our fixed effects. So in our random effect structure, we included 3 key components. So we first of all included the genetic effect of dam ID. So this was to determine whether certain dams had a genetic propensity to produce more chicks than other dams. It also allowed us to calculate a heritability estimate associated with chick production to see how responsive this would be to selection. But we also wanted to determine whether there was an environmental effect of pen ID as well. So this would account for differences in potential social structures among different pens, which could lead to varying levels of productivity. It was important that we distinguished these environmental sources of variations from the genetic ones. And then we also just included an indirect genetic effect of sire ID for completeness. And this was to determine whether certain males made some females lay more eggs and whether there was genetic variance in this trait. So there were our 3 components of our random effect structure, and now I'll go through the fixed effect structure. So the fixed effect structure of the model was to determine how different mating behaviours affected dam chick production, and we did this by using a series of models with increasing complexity. So the first model was an intercept-only model. We didn't include any fixed effects, we just included our random effect structure. And so this is so we could estimate our sources of genetic and environmental sources of variation without accounting for any mating strategies. In the second model, we included a fixed effect of the total of dam mates. And this was to account how female polygamy or female polyandry may impact female reproductive success. And in the third model, we extended this to include our measure of chick paternity. And this was to determine how different levels of male monopolization impacted female reproductive success. And then finally, in the fourth model, we looked at how the female's primary male mate impacted her reproductive success. So if we just start with the results of the first model, we examined the environmental and genetic effects impacting dam chick production, and then for each variance estimate, we also have repeatability and heritability estimate depending on whether it's environmental or genetic. So looking at the environmental effect of PIN-ID, there was a strong environmental effect of PIN-ID, indicating that social interactions in the pen were influencing the number of chicks. And it had a reasonable repeatability of 0.25, so that 20% of this variation could be down to some kind of social structures within the pen. We also had a reasonable estimate for the genetic effect of dam ID as well, showing that some dams had this genetic propensity to produce more chicks than others. And then finally, we also found an indirect genetic effect of sire ID. So this suggests that certain males were influencing females to produce more chicks than others.
Can I ask something? So
Yeah.
the one that you call repeatability is actually for the environmental effect, is actually the common environment. It's not really a repeatability, is it? So the repeatability should be the animal component plus the permanent environment due to the animal, because yes, repeated measurement. But the effect of the pen looks more like the common environment.
Yeah.
So
Maybe this is a question of terminology. What would you suggest in that case?
yeah, I mean, looking like this, I think it's the common environment.
Okay, common environment. Yeah, okay.
Yeah, an effect, but
Yeah. Yeah, because I suppose repeatability is an estimate of an individual, usually, whereas
yeah,
maybe this describes what you're saying. Yeah, thank you.
Actually, as well, so that the effect of sire ID, I mean, in a funny sort of way, it seems a bit strange how it influences the number, but of course, this is just chicks hatched, isn't it? You don't know. There's no. Well, I guess that is partly why you're trying to do the next thing. It's to know if it's the fertility, as it, is there some environment, embryonic lethality or something?
Yeah. In this, we can't really separate that. It's just all the data we had on this project was just chicks hatched. Data recorded on any chicks which failed to hatch, it just doesn't exist at the moment.
Yeah.
Okay. So model 2, we looked at the effect of total dam mates on female reproductive success, and we predicted that a higher rate of female polyandry would lead to an increase in female chick production. So do female chicks which have more mates, do females which have more mates, do they produce more chicks? The answer is yes. We found that the more mates a female had, the number of chicks she produced increased. So this confirmed part of our hypothesis that females could overcome sperm limitation by just mating with a greater number of males. I'm just going to skip over this bit because I want to get on, but it's just more variance estimates. Number 3, we additionally looked at the impact of male monopolization on female reproductive success. And here we predicted that females which were going to be more monopolized by a male would produce fewer chicks. So what did we find here? So do monopolized females have fewer chicks? So on the x-axis, we have skew in chick paternity, where higher values of skew indicate higher levels of male monopolization, and lower values indicate that paternity is more equal amongst males. And then on the y-axis, we have the number of chicks hatched per female. And actually, you can see there's a positive between these two variables, not our expected negative. So actually, what we found is that when there was a higher proportion of females brood outside by a single male, she actually increased her reproductive success compared to when paternity was more equal. So it doesn't seem that females are really suffering any negative consequences of so-called male monopolization.
So are you observing for the number of matings?
This is just a pedigree, so it's just the result of successful matings.
Okay, I was just thinking for a chicken, there's usually the dominant male spends a lot of time mating the females to make sure he's the only one mating. So sort of,
yeah, we just have like the outcome data provided by the chick, which has resulted it. So if there were unsuccessful matings or repeated matings, we don't really have that data at the moment. Yeah.
I've forgotten, you told us near the beginning how the skew was produced, but is it in some way the values with the greater level of skew are those chick dams that have more eggs or more hatched eggs, because
so we
the
tried
greater number that it lays, the greater the chance of observing more sires, perhaps, having produced those eggs. If you get my, I mean, if it's only produced one egg that's hatched, there's going to be no skew, and even with small numbers, it might just by chance be the same male. But as you increase the number of hatched eggs or fertile eggs, maybe, in other words, is there any correlation between number of eggs laid and the skew?
So we do have an interaction between those two measures, which I'll column onto. But just to kind of highlight that we chose this measure of reproductive skew because it didn't correlate obviously with any of our other fixed effects that we wanted to include, which would have sort of confounded some of those questions we were trying to ask.
No, I just wondered.
Yeah, no, I think it's, these were questions we were asking all the time.
So how do you actually, you say that female monopolization produce less, it doesn't have an effect on the, female monopolization produce higher amount of chick, but at the same time the number of mating that the female has with higher number of chick as well. This seems to be a bit contradictory.
Yep.
Yeah. Is it because model 3 doesn't include another? Does model 3 includes the fixed effect of number of mate as well?
Oh, and I completely agree. They seem contradictory results, and we were scratching our heads over it. We were like, oh, these are showing a really similar effect. What's going on? How can we parcel it out? So yeah, we included a interaction term between these two, which I can explain, which may have kind of help to kind of answer some of those questions.
Yeah, but I was just wondering what is the you show the result for number of the effect of the number of mate for model 2, but for model 3 it had the same trend.
Yeah. So when we increased the complexity of the model, we still had that same effect that we were observing in model 2. So yeah, things didn't really change. So in the fourth model, we looked at the effect of male polygamy on female reproductive success, and we predicted that females that mated with more males would have fewer chicks due to sperm depletion in the males. So the question here was, do sperm depleted males cause females to produce fewer chicks? And there wasn't really a great deal of evidence to show that they did. The more mates that a sire's primary, the more females that a sire mated with, the number of chicks which are hatched for a particular female didn't really change very much. So it didn't seem that sperm depleted males were causing females to produce fewer chicks, is essentially what this result shows. So as we found an effect of the total number of dams and skew, as we found the effect of the total number of dam mates and skew on chick paternity on the number of chicks that a female produced, we decided to run a final model where we looked at the interaction between female polyandry and skew in chick paternity to really determine what the interaction was between both these terms. So here we're asking, does female polyandry and skew in paternity impact female reproductive success? And we found a negative interaction effect, which was really difficult for us to interpret what that meant. So I plotted the relationship to try and make it clearer. And then I'm just going to talk you through the results just to show what these mean. So on the x-axis, we have skew in chick paternity, where higher values of skew mean that a larger proportion of a dam's brood is sired by the same male. And on the y-axis, we have the predicted number of chicks which are hatched per dam, which was obtained from the model. And then these coloured lines represent the different number of different levels of female polyandry. So females could here range from having 2 mates up to 13 mates. So the key observations to take away from this graph is that if we just focus on the left-hand side of the graph, females with more mates generally have higher reproductive success than those with fewer mates. But the impact of skew changes with the number of mates that a female has. So if we just focus on the black line first, this is when a female had 2 mates. Higher skew resulted in greater reproductive success for that female. If we then look at the yellow number, that's the yellow line, so that's when dams had about 11 mates. Skew seems to start to have very little effect on reproductive success. And then if we look at the blue line, this is when a female is predicted to have 13 mates. Actually, higher skew begins to actually cause a decline in the female's reproductive success. So really the takeaway from this graph is that females with few mates can actually really increase their reproductive success by having a really high reproductive skew. And females with more mates, say up to about 11, can increase their reproductive success further by ensuring their brood is sired by just 1 or a few males. So this kind of explains this negative interaction term, where females with more mates, they have a positive so it explains this negative interaction term, where the positive impact of reproductive skew diminishes as mate number increases. Okay, so was there any correlation between these, between female polyandry and skew in chick paternity? Well, we did actually find that higher rates of female polyandry were associated with more equal paternity distribution within a dam's brood. So we can sort of interpret this result in a number of different ways. So from a negative correlation. So females which mated with more males, the distribution of paternity among their broods became more even, is what we can take from this. So from a behavioural perspective, we could argue that there's a presence of two distinct behavioural strategies. One where females focus on mating with a higher number of males. This increases sperm competition, and it also reduces the likelihood that a single male will dominate paternity. But equally, the female will benefit from having increased genetic diversity in her offspring. But it does expose females to the risk of more male harassment. Whereas there can be another strategy that exists within the population, which could be low polyandry. So when females mate with fewer males, the probability of a single male dominating paternity increases. So she'll benefit from lower genetic diversity. Well, she'll have the cost of lower genetic diversity in her offspring, but will have the benefit of reduced male harassment. So what could the implications be this from an evolutionary perspective? Well, this could indicate some kind of balancing or frequency dependent selection, where both strategies are maintained within a population because they just confer fitness advantages under different conditions. And they could be also shaped by the social environment of the pen as well. But then from a breeding perspective as well, this antagonistic relationship between polyandry and paternal skew could mean that selection on one trait could negatively impact the other. So it's just worth considering that in managing selection programs. So just returning to our results, how do our sperm limitation predictions match results that we found? Well, from the female polyandry perspective, we hypothesized that females that had more mates would have more sperm available to fertilize their eggs. So we predicted that the increase in mates would lead to an increase in chick production, and our findings match that result. But in terms of male monopolization, we predicted that females that monopolized would have less sperm available, and so there would be a decline in chick production. But our findings did not match that prediction. Instead, what we found is that higher skew in chick paternity was actually beneficial to the females and then caused an increase in chick production. And then finally, we predicted that male polygamy would cause males to become sperm depleted, and so chick production would decrease. But we didn't really find any evidence of that relationship. So overall, we found quite limited evidence for the sperm limitation hypothesis. So what just could be some alternative hypotheses which could explain some of our results? Well, one potential explanation could be the genetic compatibility hypothesis. So this refers to the likelihood of producing viable offspring when specific males and females mate. And females can benefit from increased heterozygosity in their offspring through reduced risk of lethal or deleterious alleles. And this can be quite important in closed populations, such as those involved in commercial production. So female polyandry could be seen as a strategy to increase the chances of coming into contact or mating with a genetically compatible male. And our observed relationship between paternal skew and female observed relationship between paternal skew and female reproductive success could reflect the increased survival of embryos from these compatible male-female pairings. But in order to test this genetic compatibility hypothesis, we would need to collect some additional data beyond what we currently have. So we currently only have DNA collected from successfully hatched chicks. But in order to investigate this genetic compatibility hypothesis, we would also need to collect DNA from non-viable embryos as well. This is something which we're keen to explore further, and Kelly and I, we've put in an application for a BBSRC link grant in collaboration with Cherry Valley Farms, and we really want to distinguish between this infertility and embryo mortality, and this is an outcome that we're currently waiting on. So another potential explanation for our results is cryptic female choice. So this refers to post-copulatory processes where females can bias fertilization towards sperm from specific males. And females would benefit from selecting sperm from males who may provide the greatest fitness benefit to their offspring, for example. And mechanisms that may facilitate this could be females selecting sperm from their preferred males, or females trying to actually actively discard sperm from less desirable males. So we know this happens in chickens. Females will eject sperm following forced copulations. We don't quite understand the degree it happens in ducks yet. But we know in wild mallards, about an estimated 40% of matings have been observed to be forced copulations, but we're not quite sure the extent this occurs within the derived Pekin duck populations. So one way that we can investigate cryptic female choice is using factorial designs such as North Carolina 2. You can expose females to sperm from multiple males and look at the variance in both male fertilization success, variance in female success, and also the significant male-female interactions. And if we identified these male-female interactions, this would start to point towards cryptic female choice.
Do they have sperm storage glands like chicken? I assume they do.
Yeah, yeah. Like almost all birds, I think.
Yeah, I imagine so.
And then finally, there's the male bodyguard hypothesis. So mate guarding by males could actually indirectly benefit females by reducing male harassment, for example, reducing their injury and physiological stress associated with forced copulations. And so mate guarding could actually just improve reproductive success by improving the welfare of the female. And as mentioned, forced copulations can be up to 40% in wild mallards, and we know this results in injury and mortality in some of these birds. And females have actually been found to forage more efficiently when they're guarded. So this points towards male bodyguards as being a potential reason why we were seeing this relationship between skew and female reproductive success. So this brings me on to my concluding slide. So we found that female mate number and paternal skew influenced chick production. Both genetic and environmental variants exist for both female polyandry and paternal skew. This can provide targeted selection of traits, but also highlights the importance of social interactions that were occurring within the pens. It really highlights that sperm limitation is not the primary factor affecting reproductive success, but there could also be alternative hypotheses such as genetic compatibility, cryptic female choice, and the male bodyguard hypothesis. But really I just want to end with concepts from behavioural ecology can provide a framework for understanding and enhancing productivity traits such as reproductive success in managed populations. And then finally, I just want to end with, could your data benefit from a behavioural ecology perspective? So I'd be very willing to help provide a novel insights into data by helping uncover patterns of growth, feeding, welfare movement, and disease resistance and other traits. Just contact me by email. So I'm looking for job opportunities currently. So I'm available from April 2025. So just keep me updated, everyone. So yeah, thank you very much.
Thanks, Cammy. Any questions in the room here?
I was just wondering about this relationship with consistency variation that you actually assume that for the female monopolization actually increase the number of but how the coefficient variation might not be necessarily mean monopolization because you divide it by the coefficient variation divided by the mu by the mean, and if we actually assume that there's a maximum number of, there's always like upper limit of the number of eggs that you can hatch because an animal only can produce 1 egg a day, is it?
Mhm.
So divided by n by the mu that actually is the number of it's actually related to the number of male. Basically, you are actually the coefficient variation is such as basically you're fitting number of mate. So that's the reason they have the same trend. So I wonder if you actually fit the actual variance rather than the coefficient variation, it might actually change the
So we fit variance instead of coefficient of variation.
You fit, yes, that's, I
Yeah.
believe that perhaps that, because the variance actually still tell you that if there's
Yeah.
more variation, it means that somebody is getting more than somebody else.
Yeah, so
Somebody gets,
we did explore with putting that in the model, but because we were really interested, like from our hypothesis point of view, that what is the effect of mates and then what is the effect of paternal skew, we thought it was really important to have this measure of reproductive skew which could distinguish between those two effects. So whilst overall biologically we see that mate number is the biggest driver, we wanted to separate those two effects.
but my argument is not separating it because you are divided by n. So actually, if you actually plot for each individual, for each female, you plot the number of mate and plot the coefficient variation, probably you'll find a direct relationship which will be negative because of the fact is divided by mu. So you're having actually corrected by the mean, you create the actual value to be related to the number.
Okay, I can plot them out, have a look.
Yeah.
Yeah, okay.
Because you looked at all those.
Yeah.
You looked at all the different things. I suppose it's just going back to that data and just, sort of plotting,
Yeah.
why you chose not to do that one.
Yeah.
There was a good reason.
There was a good
I just don't
reason.
remember
I mean, I feel like I've tried to explain the reason, but I don't think maybe it's coming across clearly enough.
and you had a baby in between time, so that
Yeah.
sort of skewed the length of time.
Yeah.
Are all the males and females the same age? They all hatched at the same time?
No, there's quite a bit of variability just due to Cherry Valley's production process. So, I mean, depending on who ends up in what pen can just be quite variable depending on what they've done at the time. So that could be a big factor.
Maternal condition as well. I'm wondering if
Yeah.
maternal condition babies, and that might affect reproductive strategies as well.
Yeah.
in females.
So
Do you have that data? The Cherry Valley have that data, like age, weight, and like that?
yeah, so they have some additional data which could be fitted as covariates. Previously, when I've looked at other behavioural measures and tried to put weight and an age to them, I haven't seen much of an effect. But yeah, in this case, we were just quite interested in mate number and kind of almost putting all those other effects into that environmental component.
questions. Mino.
Mino, do you have a question?
Hi. Yeah, Cammy. Yeah, nice presentation. And I was thinking that did you also check in your data the I think you posted in your before conclusion slide about the male variants. So how much fertile the male ducks were actually?
Yeah, so we also looked at all of those 5 models that I presented, but from the male perspective. And we found a very similar trend from the male perspective as well. So I think that sort of goes to show that it is sort of driven perhaps more by this genetic compatibility type of explanation.
So if you already have on per male how many chicks were there, and if you put them do the reverse modeling on the male side, keeping the female IDs as a indirect effect to see, because maybe your skewness is only coming from that rather than incompatibility, or also because of the behavior, because they are more in wild are monopolist rather than polyandrous.
So we did all those reciprocal models. I would have to go back and check and look at what results I got in the interpretation, but I can't think of them off the top of my head. But from recollection, it was quite a similar story from the male perspective.
Okay. Yeah, you already checked, so it's already great. Thanks.
One comment and one question, one opinion.
At least you're honest about the comment.
So the comment, just following on from Liz's comment, question was that the range in terms of age is about 12 weeks. And the real impact is the level of maturity when the birds go in. So although they all go in at a relatively similar condition score, we were interested in what they came out at, you know, whether sort of declining condition versus birds just getting really fat in there as a, you know, they're meat type birds. So that's just something that we were interested in looking at. And I think that Cherry Valley started to collect that outweighing data. But I don't think that we had time to explore it during this project. The question is, that 1:4 ratio, do you think that's an important component? Because obviously in chickens, it's more like 1:10, 1:12 in commercial systems. So do you think that Cherry Valley have it wrong, and whether there might be something different if we had something a little bit more extreme in the pens?
Yeah, like it's interesting with the 1:4 ratio, because I'm not quite sure where it's come from. It's just the ratio that they use, and there's not an opportunity within their commercial system to kind of experiment with that. So that's something that we would have to do within Roslyn, I think, to get down at that.
It is odd though, because certainly in the chicken world, I mean, some effort was put in a while ago to make sure it worked and was optimal. So I find it hard to believe somewhere in the past somebody hasn't done that, but that seemed quite a lot.
Yeah,
lot of
Yeah.
Well, we have
knowing what they're like.
Yeah, we kind of hear is, you know, that the 1:4 ratio works for them, so that's what they go with.
But isn't that more of the natural structure? So they form, donkeys, ducks form monogamous pairs in the wild.
It's actually
long term.
It's actually quite interesting. In the wild, you have the reversal of this mating pattern. So I'm not quite sure the evolutionary reasons for it, but you have a bias towards high number of males in the population, and this is why you get a lot of coercive mating attempts by males. So if you're not in a stable relationship with a female, the only way that you're going to be able to secure your matings, your successful matings, is to use more coercive tactics. And that's when you see sort of females getting sort of attacked at local ponds. As I say, I'm not quite sure what is driving that imbalance in the sex ratio. But it's interesting when you come into commercial population, they reverse it. So yeah, it's things to be looked at, I think.